So while Sudhir is coming down here, Sudhir was one of the original crew along with me when Biodesign opened. And Sudhir was really the king of the computer analysis of genomics and has become ever more so. He was at Biodesign, from 98, or ASU, 98 to 2014. And then Temple University said you can have your own center, right?
Yes, indeed, yeah.
And he fell for that. I hope he hasn't regretted it too much. So he's currently the Laura H. Carnell Professor and Director of the Institute for Genomics and Evolutionary Medicine at Temple University. And I just want to drop one little statistic. The last time I looked, Sudhir's citations were over 300,000. Sudhir.
Thank you. Well, with citations, it's always you who did it, not me. So I appreciate everybody's appreciation of this statistic. And it's a currency for people who develop methods and tools. It's not by itself any more important than what everyone else is doing. So today what I would do is I'll talk about my research program, because this is what Stuart's emails seem to suggest. Ever since I began my career in academia, starting at Penn State. And Biodesign, when it was first envisioned with Charles Arnson and after that many other wonderful leaders, was a special place for discovery and translation at the interface of sciences and engineering. To me, this was like a perfect thing ever to happen, because I had, by chance, without thinking about it, done degrees in biology, genetics, and engineering during my undergraduate and graduate years. So to me, this was like the catnip, So somebody said, oh, but Biodesign requires that you have certain amount of funding. I thought, well, they could kick me out, but I'm going to go in and have fun. And I think over the next 14 years almost, or 12 years, I had a lot of fun in Biodesign Institute. And part of me got me away from wonderful Arizona to really Philadelphia, which I also like, since I come from an old city, Delhi, to another old city, which is just Philadelphia. But what I will do today is just present a few highlights of my career, starting from my graduate career, just show you like how things progressed, why I did what I did. I don't really have a cool story like Parag's in the morning. I had no confusion. Actually, I had no knowledge. I don't even remember what I was doing at age 18. Except my father said, you be an engineer or a doctor so you can feed your family. I said, all right, I'll try. And that was a simple life. So I really have great empathy for Parag, having gone through so much. So what I do is mostly need-inspired or use-inspired, you can say. Being an engineer, I'm interested in solving problems. Being a biologist, I'm interested in finding problems that can have solutions and hypothesis testing. So what I do is molecular data-driven discovery. And in that case, I'm interested in developing methods and tools, building software, packages, for example, analyzing big data sets to look for patterns that are interesting, and then harnessing them for translation if possible. And my main raw material for studies are mutations. And the mutations can be very recent, like in coronaviruses, or they could be quite old, like millions of years ago, and in between, for example, tumor variation that arises over a lifetime, inherited variation that causes disease that arises over populations of humans. I'm interested in all of these because fortunately, using the principles that we use, you can study all of them and you can connect them all together. So I'm going to just start with this part, which is millions of years. So when I came to graduate school at Penn State, basically we were comparing sequences between species, primarily. I did not want to do population genetics because there wasn't that much data at the moment. It began to arise more and more, but my mentor, Masato Shinei, was very interested in molecular evolution, that often related to species and genes differences. So that's what I did. So being an engineer and a biologist, as soon as I came to the lab, I realized there were no tools which were user-friendly. There were a bunch of command line tools that biologists at that time were like very afraid of. They thought that was the worst thing they could do. But so they didn't have any tools to apply sophisticated methods to analyze sequence data. So I said, I didn't have one either. So I made one for myself, and my advisor said, hey, this can be chapter 1 of your thesis. Why don't you make it for everybody? I said, all right, what's a big deal? So I made a user interface, and it wasn't easy because the amount of memory on a personal computer in 1990 was 640 KB, kilobytes. Kilobytes. This is not megabytes or gigabytes or terabytes, it's kilobytes. So you're working with this little bunch of morsel of the memory, and you have to implement these algorithms to build phylogenies or estimate things, visualize in this sort of a character-based display things, and design the interface so everybody can sort of follow along because it has to be intuitive. So essentially, I did that not knowing that what I did was good, because when you work on these things after going to a bar, arrive back in the room at 2 a.m., you can't do anything complicated. So I kept everything simple, and it seemed to be the trick. So now you know alcohol can play a good role in your life. So we just distributed that software in diskette. There was no internet that you can send stuff, no websites. And so this was the diskette I produced. So I was a production manager too. And people will send us letters to request the diskette and a manual, and we will ship it by mail. So this 2,500 of them we distributed. This tells you how much need there was at that time for something like this, which I did not know, of course. I just came in, fell off the boat from India. I mean, I have no idea. So I did this, and let's say I got lucky, right time, right place, with right ability. And so that happened. So then I arrived at ASU in 1998. Maybe I got the job partly because I wrote Mega. Now a number of people at ASU were using it. I learned afterwards. But by the time I came to ASU, I had stopped working on Mega after 93. By 98, more than 300 people were citing Mega every year, which was kind of weird for me. My PhD advisor was very much into citation counting. So he'll go to the citation index and use a foot scale and count how many every year. He's an amazing scientist. I mean, got a Kyoto Prize and everything. But he was citation happy, and so I am too. You inherit that. But then the data started to increase from 50 million bases in GenBank to 50,000 million bases. And so you got bigger data sets. Even at that time, these were not really big data sets from today's perspective, but from then, they were big, faster computers. Graphical interfaces become much more widely available, like Windows 95, kind of an ugly Windows 95. And so I wrote my first R01 from Temple, saying I want to develop the next iteration of Mega and some methodologies. So we developed Mega 2. And we had to innovate new methods, faster algorithms, new interface. This took years to do, but ultimately we published it. As you can see on the right side, the second set of displays look better than the first ones to some extent. But if I had not made Mega 2, Mega 1 would have died a happy end. As you can see, 98, 99, 2000, it was flat. People were not using these kind of programs. They wanted more sophisticated programs. So by doing Mega 2, unknowingly, I rescued Mega. And then once we published that, it went fast. A lot more people began to use it. And at this time, when I was leaving Penn State a couple of years before, people wondered why I left Penn State. And I said, because I was a student. Because they had not realized that a graduate student in their first year could write a program that many people will use. So I will say, be adventurous and just write what you want and do what makes you happy. So doing this stuff made me happy. It was my hobby, actually, but turned into a lifelong passion. So Mega over time has evolved until Mega 5 in 2011. It was at ASU in Biodesign Institute. We had exceptional staff and programmers, which helped us move forward by developing many different versions of Mega. Mega 12 is what it looks like now. You can see on the left side here. And on the right, you see the number of cumulative citations, the number, that 300,000 that Stuart just mentioned. That's because a large part of it is because of Mega. It's 240,000 citations to Mega. Every year, 20,000 people cite Mega, which is something you can't comprehend, I can't comprehend, and I'm grateful people are interested in it. But half of those citations were actually for the software that we developed at Biodesign Institute. And so it was a big part of our success. And people use comparative sequence analysis and phylogenies in essentially every part of biological science. As you can see from the right panel, it's in every biological field that exists there. So this gives you an idea that, as a graduate student, you do something without really knowing what you're doing. But sometimes this is exactly what everyone needed. But the idea was that there was a real clear-cut need. Doing incremental research is really much worse. So you rather do something brand new that no one else has done and take a risk, and maybe it'll work out. So what's driving Mega 12 now? So so many people use these software packages that align sequences, that build phylogenies for big data. They consume huge amount of time and memory and produce, greenhouse gases. So one of my passions recently has been introducing green computing, in which we want to develop methods that take fewer compute cycles, less memory, and are big data friendly, and could actually be used on a personal desktop. And this is while maintaining the accuracy. So this is a hard thing to do. We have developed many such methods, but this is possible. And that's where I think revolution is, where you have to develop innovative heuristics, analytics with fewer assumptions, and even AI. So we have now implemented AI methods in Mega. The benefits are that people in third world countries, or so-called third world countries, or second world countries, or third world laboratories in the US can actually now do big data analysis on desktops. Otherwise, they had to do HPC. You spend so much money, and you throw away old computers that are probably still very good. So in Mega, the green computing is our interest to bring the ability to do analysis for big data to desktops. So that's what I'm requesting NIH for more money to really simplify life for a lot of people, which will be, I think, quite enriching. So this is the second animation that is not working, something wrong with the animation. But my second highlight I'll talk about is time tree of life. We take a molecular data-driven approach to building a tree of life scaled to time. Again, it began at Penn State, not a part of my thesis, PhD work. I graduated in 1996. I was doing this on the side with my colleague Blair Hedges, who was on the 6th floor. I was on the 3rd floor. And we downloaded all the data from GenBank. It was possible to do because it was so little. And we just found all the genes we needed for every species we needed. And this is like data-driven discovery with full data. So this is big data analysis. Today you will call it like minuscule, but at that time it was big. And we would estimate the divergence time of humans and mice. So there was a prevailing theory that dinosaurs went extinct and then major groups of mammals emerged. And that happened around 60 odd million years ago. Well, we said, well, let's see if molecular data agree with this conclusion from the fossil record. Because fossil record have one problem, which is the presence of fossil record is good, absence, we don't know what was really there. So we estimated that this time was around 90 million years ago, not 60. And it predated dinosaur extinction significantly. So we were there as major groups like proto-primate, proto-rodent at the feet of the dinosaurs. So then the question arose, what could give rise to these major groups of mammals? And we posited the hypothesis that it was continental breakup, that Pangea was breaking up, and by 90 million years ago, it has really become a large number of land masses. And in fact, this thing has proven to be correct now in many studies, where now you can actually pinpoint which particular mammal group, major group like Afrotherians, elephants and others, where they arose, which particular landmass, like Afrotherians arose on African landmass. And then, we did another bigger one where we took all the vertebrates, downloaded all the data. So you can do these things without telling your advisor. And I did, however, asked him to be a co-author on both the papers. He said, nope, you've done it, you deal with it. So I dealt with it. But he was very happy with Mega, I think, so he let me get away with a lot of weird things. Uh, so, so then what happened is a lot of people got very interested in dating species divergences, and many, many studies started to accumulate in the literature. So this is a big problem for everyone like you guys and sometimes even me, and particularly general public. What is the divergence time between human and cat and dog? You have to go read through these really boring papers. And believe me, that is an area that you don't want to get into. So we thought we need a database and a system that can actually automatically give you the answer you want using all the available data. So we started to curate published results, which has become the new type of data, and do meta-analysis, and then build a database where you can find this information. So you never have to read any papers. That's the main point. We remove the paper reading problem. And then this also was interesting to us because we wanted to synthesize a global time tree of life, which we could not do since every study has some sort of species, but not all species. So we wanted to do all of this. And so at Biodesign Institute, in year 2006, we published the first time tree database, where you could go in and say, I want cat and dog divergence time, and it'll give you a list of actual time, a median time, and all the studies that were published that we had put in our database. And our database was a relational database of trees or phylogenies. So it was very complicated, but ultimately it synthesized the knowledge in a very effective manner. And so then we also made it for the iPhone, and of course there's a bunch of stories about how people were unhappy, particularly certain religious groups, that Apple allowed Time Tree app. I don't know why, but anyway, that's how it was. And then in 2009, we actually built a first family time tree of life with 1,600 families, which then David Attenborough asked us to use for his Rise of Animals series, where and I was shocked to see how they used it. They just asked us, can we use it? And they made this beautiful animation, and they show you where the lineage is traced. So we were really happy that something like this could get into the public, so they can understand evolution by experiencing it, rather than just being told it's true. And then in 2015, we published a time tree of 50,000 species, which is the largest time tree ever assembled. And it looks really pretty. We were so tall that we had to wrap it. So this is a wrap time tree. And actually, I brought some of those posters with me. If you're interested, I can give you one during the break. And the interesting thing for us was in this time tree to ask the question, how long does it take for two species to become distinct, or two groups of organisms to become distinct, the time to speciation? And by having this large time tree, we were able to do this for vertebrates, for arthropods, and for plants. And we found that this time is between 2 to 3 million years. It's not like 2 and 20 or 1 and 15. So this was very interesting observation that hasn't been disputed yet. And what it shows is that speciation in all these groups proceeds sort of like at somewhat constant like rate. Now there's a huge variability, but the mode is very similar. So this sort of goes against the adaptive niche hypothesis, where adaptation creates new species. I think adaptation might come later, but when the two species are separated for a while, they accrue genetic incompatibilities, and they just can't no longer mate. And then once they become distinct species, then they can sort of adapt to their environment that they happen to be in. So adaptation first versus adaptation later hypothesis, our results are more consistent with adaptations later than adaptations first. Now we have a new tree of 150,000 species. NCBI only has 450,000 species with binomial nomenclature. We have a third of them, and we are on our way to cover as many as we can. And now you can also actually get time trees. So if you're interested in time trees, you don't have to read any of the literatures that we had to read. And you can become very knowledgeable very quickly. And it's been cited a lot. There are more than 250,000 queries. It's widely used in teaching and many other areas. So this is another use-inspired product that we made, which also has scientific and fundamental research applications. And then the third highlight I want to talk about is very quickly is the phytomedicine area. So this is the area that I began actually at ASU. And here I was interested in harnessing nature's design or nature's information or nature's experiments that give rise to current sequences. So when you compare the sequences to each other, you find the parts that are diverged and the parts that are highly conserved. So these, I think of them as two lenses in a telescope. And then with these two lenses, you can peer into the constellation of millions of variants that you see out there in the databases of SNPs. And so I sort of proposed this phytomedicine word so that we can sort of distinguish this as a phylogeny informs medicine, not as much as we're just doing phylogenetic information, generally for medical data. And so this was sort of an area that I stumbled upon after a postdoc came to my lab who wanted to do more PopGen, and I didn't want to do PopGen. So I said, okay, let's do in between. You analyze population variation on disease and see if it is more common at variable sites or constant sites over evolutionary history. In retrospect, it was all obvious, but at the time, no one had ever thought about doing a study to find it. And so we were the first ones to show that highly conserved positions actually contain more disease variants than variable positions. And then that formed a basis for a machine learning method we developed to diagnose disease variants whose accuracy is very similar to you have in AlphaFold or alpha missense. It's just that, they're all 90% accurate. But then we developed another method which doesn't require any training data, just like large language models don't require training data. So anyway, that's another method. Again, these methods are not widely used because there are so many other methods and there's a big crowd of them. But essentially, exclusively evolutionary information is all you need to make good predictions, generally. So that is the phytomedicine where we have variants that we have acquired at conception, and there is a geological timescale that's informing us about this variation. What about personal timescales? So personal timescale is the second part, which is when after we are born, our somatic cells also evolve and diverge, and particularly high turnover cells like stem cells. And so we began asking, can we use phylogenetics for this purpose? And the reason arose is that the stem cells that give rise to blood actually constantly evolve at a pretty constant rate, as you can see in this picture. And if you look at the people with blood cancer, like MPN, they also still evolve at the same rate, almost. If you look at this picture here, they're similar. So a person has MPN or not have MPN, you can tell based on the amount of variation accumulated in their hematopoietic stem cells. So which is basically different from the incidence of MPN. It really shoots up after the age of 60. I'm getting close to it. It's going to be very problematic. So the incidence really increases after 60, but the rate of accumulation of mutations remains pretty constant. So we thought we would do a phylomarker for this to explain this incidence increase. So as you can see here, that in the infant, we have a bunch of diversifications. At 20 years, diversifications have declined, and instead you have lots of asymmetrical cell divisions, followed by more cell divisions and some changes. So what we saw that the phylogeny shape is aging. So we thought maybe we can measure the phylogeny age change, and we use ecological measures for it, and see what happens. And when we developed those two new measures, you can see the phylogeny age, let's call it, really perfectly correlates with incidence of MPN. So phylogeny decays in some way. It predicts where you are. So basically, you can estimate age of your blood in a phylogenetic sense using this kind of a model. So we were able to do this one. And what we found is that if you take individuals who have MPN, their blood ages, phylo ages, were uniformly, except for one case, much greater than their actual age. So essentially, their blood is aging much, much faster when it comes to phylogeny than their personal age. So this could be a diagnostic in the future. Of course, single-cell sequencing used there is not possible to do. So we have applied and made a new approach using bulk sequencing data, applied this to TCGA, and as you can see, all the patients are, irrespective of their chronological age, have much older phylo ages. So this is another way to do physiological aging of body, in this case, the blood, which could be useful because if you do this every year, you might see that there is an escalation in age. Maybe there will be ways in which we can address the problem. And so I don't want to go here too much except telling you that we are also studying tumors in general, the sequence variation, and building a lot of tools for tumor phylogenetics. But I want to conclude by saying that when I began graduate school, people doing analysis like me were secondary, and people generating data were primary. Fortunately, the primary people have generated so much data that we don't need you sometimes. So now the tables are turning, and in some areas, not every area, of course, and fortunately, genomics is turning a lot. So knowledge we gathered over the last 20, 30, 40 years can really tell us a lot. So I think the future is going to belong to data-driven discovery and validation, of course, in the laboratory. And as data scientists, those who are interested in these areas will see lots of need to develop new methods and tools. And ultimately, I feel that sustainable end-to-end solutions are the way to move forward in research and development. And I want to acknowledge my colleagues at 3 major universities where I've been, and collaborators who've been with me for 30 years working on various topics, and funding agencies and team members. Thank you.
Thank you, Sudhir. I hope you can convince the Bitcoin community to apply green computing to Bitcoin mining.
Well, no, Bitcoin is green, greenback, a lot of money. So it's already green.
Questions? Time for maybe one. Josh. Oh, yeah, you need
So a lot of people have talked about these metabolic markers for actual age versus, apparent year.
Sure.
Yeah. So have you compared the phylogenetic age to those metabolic markers? Do they track together?
So the phylogenetic age data that we are using, they don't have really much other information at the moment, so we couldn't compare them. But there is a tremendous amount of work on CPG clocks and physiological clocks. There are even companies, as you know, that actually will tell you tissue age for you with different tissues. But they all tell you, well, you're 45 chronologically, but you're 43 or 46. So they're not so effective yet. So I think that in the future, we hope to do this kind of analysis. But data are incredibly sparse in this because people haven't really sort of done things in this way. Right. I think a lot of people are still working on the old style, but some of the authors whose work we used, data we used, are really at the cutting edge of doing cool things. So yeah, that's what we will love to do, is to do other metabolic markers for these individuals.
Okay, thank you. Let's thank Sudhir again.
Thank you so much.